physics and machine learning to apply cutting-edge techniques to predict the 3D structure of a protein based solely on its genetic sequence. Our system, AlphaFold, which we have been working on for the past 2 years, builds on years of prior research and using vast genomic data to predict protein structure. The 3D models of proteins that AlphaFold generates are far more accurate than any that have come before, making significant progress on one of the core challenges in biology. What is the protein folding problem? Proteins are large, complex molecules essential in sustaining life. Nearly every function our body performs, contracting muscles, sensing light, or turning food into energy, can be traced back to one or more proteins and how they move and change. The recipes for those proteins, called genes, are encoded in our DNA. What any given protein can do depends on its unique 3D structure. For example, antibody proteins are Y-shaped and are akin to unique hooks. By latching onto viruses and bacteria, antibody proteins are able to detect and tag disease-causing microorganisms for extermination. Similarly, collagen proteins are shaped like cords, which transmit tension to cartilage, ligaments, bones, and skin. Other types of proteins include CRISPR and Cas9, which act like scissors and cut and paste DNA. Antifreeze proteins, whose 3D structures allow them to bind to ice crystals and prevent organisms from freezing. And ribosomes that act like a programmed assembly line, which help build proteins themselves. Figuring out the 3D shape of protein purely from its genetics is a complex task. The challenge is that DNA only contains information about the sequence of a protein's building blocks called amino acid residues, which form long chains. Predicting how those chains will fold into the intricate 3D structure of a protein is what's known as the protein folding problem. The bigger the protein, the more complicated and difficult it is to model because there are more interactions between amino acids taken into account. As noted in Levinthal's paradox, it would take longer than the age of the universe to enumerate all the possible configurations of a typical protein before reaching the right 3D structure. An animation of the gradient descent method predicting a structure for CASP13 target T1008. Alzheimer's, Parkinson's, Huntington's, and cystic fibrosis. We are especially excited about how it might improve our understanding of the body and how it works, enabling scientists to design new, effective cures for diseases more efficiently. As we acquire more knowledge about the shapes of proteins and how they operate through simulations and models, it opens up new potential within drug discovery while also reducing the costs associated with experimentation. That could ultimately improve the quality of life for millions of patients around the world. An understanding of protein folding will also assist in protein design, which can unlock a tremendous number of benefits. For example, advances in biodegradable enzymes, which can enable protein design, pollutants like plastic and oil, helping us break down waste in ways that are more friendly to our environment. In fact, researchers have already begun engineering bacteria to secrete proteins that will make waste biodegradable and easier to process. To catalyze research and measure progress, What happens next? The success of our first foray into protein folding is indicative of how machine learning systems can integrate diverse sources of information to help scientists come up with creative solutions to complex problems at speed. Just as we've seen how AI can help people master complex games through systems like AlphaGo and AlphaZero, we similarly hope that one day AI breakthroughs will help us master fundamental scientific problems too. It's exciting to see these early signs of progress in protein folding, demonstrating the utility of AI for scientific discovery, even though there's a lot more work to do before we're able to have a quantifiable impact on treating diseases, managing the environment, and more. We know the potential is enormous, with a dedicated team focused on delving into how machine learning can advance the world of science. We're looking forward to seeing the many ways our technology can make a difference. This work was done in collaboration with Richard Evans, John Jumper, James Kirkpatrick, Laurent Sifri, Tim Green, Chong-Li Kin, Augustine Zidek, Sandy Nelson, Alex Bridgeland, Hugo Penedones, Stig Peterson, Karen Simonian, David Jones, David Silver, Corey Kavukuoglu, Demis Hassabis, and Andrew Sr. DeepMind.